So in this video, I'm going to tell you a little bit about the genes that are on the plasmid for the pGLO lab. I'm going to take you through the different components and then how they're regulated. The first thing that we'll see is the yellow bit on the plasmid, and that is the ORI. That's the origin of replication. And that piece is needed in order for the bacteria to replicate that plasmid. And if that piece were not added to the plasmid, then when the bacteria replicate, they would not include that plasmid during the replication of the DNA. The second gene that we see is added to the plasmid is called the AMPR gene, and that's the gene that codes for an enzyme called beta-lactamase, which is an enzyme that can break down certain antibiotics. And in our case, the antibiotic that we used was ampicillin. So beta-lactamase is going to break down the ampicillin. The activity of that gene is controlled by what's called a constitutive promoter. And that is a promoter that occurs at the beginning of the gene. And constitutive tells us that that promoter is always turned on. So if the promoter is always turned on, then that means RNA polymerase is able to transcribe that AMPR gene at all times. And we have another gene that is also under the control of a constitutive promoter, and that is called the ARA-C gene. And I will tell you a little bit about the ARA-C gene in a second, but the fact that the ARA-C gene is under the control of a constitutive promoter tells us that the ARA-C gene will always be transcribed, and that means that that mRNA for the ARA-C gene will always be translated into ARA-C protein. And then the third gene that we have on the plasmid is the GFP gene. And unlike the other two genes that we saw were under the control of a constitutive promoter, the GFP gene is actually controlled by what's called an inducible promoter. And an inducible promoter is a promoter that is not always turned on, but instead it has to be turned on by the presence or absence of a regulator protein. So I will walk you through how the GFP gene is regulated through the activity of that inducible promoter. Okay. So all bacteria, or I should say many bacteria, have a plasmid that is under the control of what's called a pBAD promoter. And a pBAD promoter is a promoter sequence that occurs before the presence of three genes that are within an operon. And the operon's name is called the Arabinose operon. And the Arabinose operon contains three genes. The three genes are ARA-B, ARA-A, and ARA-D. And when those three genes are transcribed and translated, they result in the production of proteins or enzymes that are important for the bacteria's ability to use a sugar called arabinose as a food source. So the pBAD promoter is an inducible promoter, and that means it will only be turned on, and RNA polymerase will only be able to transcribe ARA-B, ARA-A, and ARA-D when it needs to. And a bacterium might need to activate the pBAD promoter and transcribe those three genes in the Arabinose operon when it does not have access to glucose, and it does have access to arabinose. So if we add arabinose, well, I'll take a step back. Most of the time, the pBAD promoter is turned off, which means the ARA-B, ARA-A, and ARA-D genes will not be transcribed or translated. And the way that the pBAD promoter is turned off is through the presence of this protein. It's called a transcriptional regulator, and the protein is called ARA-C. And that ARA-C protein, when it binds to the pBAD promoter, it blocks the RNA polymerase from binding to the pBAD promoter and then transcribing ARA-B, ARA-A, and ARA-D. But in the presence of arabinose, which is sugar, the arabinose will actually interact with the Ara-C protein, and it will change the shape of the Ara-C protein, and it will cause the Ara-C protein to fall off of the DNA. So if the Ara-C protein falls off the DNA, now the PBAD promoter is accessible by RNA polymerase. So RNA polymerase will bind to the promoter site, and then as it reads the DNA, it will transcribe the ARA B, A, and D genes into ARA B mRNA, ARA A mRNA, and ARA D mRNA, which then can get translated into their respective proteins. So what scientists have done to form the plasmid that we used in our pGLO lab is kept most of the parts of the system the same, except what they did was replaced the ARA B, ARA A, and ARA D genes in the DNA on that plasmid with the GFP gene. So they kept the PBAD promoter as it is, but they replaced those genes with the GFP gene, so that the GFP gene could, the expression of that gene could be regulated by the presence or absence of the sugar arabinose. And so we'd expect that if arabinose is present and the PBAD promoter is no longer being inhibited by the Ara-C protein, because the Ara-C protein and the arabinose have fallen off the DNA, that we are able to allow RNA polymerase to bind to the PBAD promoter and then transcribe the GFP gene into GFP mRNA, and then the GFP mRNA will be translated into GFP, the protein, which is the protein that allows the bacteria to glow green. So in this lab, what we did was we inserted a plasmid into the bacteria, and this plasmid was constructed specifically to allow us to control the regulation of a gene on that plasmid called the GFP gene. And the regulation of that expression was under the control of a PBAD promoter. And that PBAD promoter is going to be activated when an arabinose sugar is present in the petri dish. And as a result of that PBAD promoter getting activated, the GFP gene is transcribed and then translated into GFP protein, and the bacteria will glow green. So a quick summary of that information is shown here, where we have the Arabinose operon as it normally appears in a bacterium on the left side of the screen, and then what that would look like if we took the ARA B, ARA A, and ARA D genes and cut them out of the plasmid and replace them with just the GFP gene. So I would take a screenshot of this or write down what's happening, because the regulation of GFP is a fairly important one to understand, at least for the post-lab questions and for how this is used in bacterial transformation in general.